Forget plaques. The real party in your brain might be happening somewhere else entirely. While researchers have spent decades staring at amyloid plaques and tau tangles under microscopes like crime scene investigators, new science says we might have been ignoring the louder, messier culprits, the lipids. Yes, your brain lipids are having a party, and not the good kind. Inside your brain live microglia, the immune cells that usually act like no-nonsense bouncers. They patrol the place, clear out dead cells, and keep amyloid beta, the sticky protein that causes plaques, from piling up on the dance floor. But when things go wrong, these bouncers stop working the velvet rope. Instead, they get distracted by all the fat building up inside them, like someone at a party who finds the snack table and never leaves. Suddenly, they're too busy hoarding lipids to keep clearing debris, and the result is that plaques and inflammation spiral out of control. Researchers at Purdue University recently showed that this fat accumulation isn't just a background effect, it might actually be driving the disease. They found that microglia closest to amyloid plaques are stuffed with twice as many lipid droplets as those farther away, and their cleanup ability drops by almost 40%. This means the more fat drunk they get, the less work they do, creating a vicious cycle where plaques grow, inflammation rises, and microglia keep stocking up on more fat. Instead of acting like professional immune cells, they turn into couch potatoes surrounded by greasy takeout boxes. But here's the twist that makes this research so exciting. The fat party can actually be shut down. The team traced the fat-making process all the way to its last step, which is controlled by an enzyme called DGAT2. Think of DGAT2 as the DJ who keeps the party going by pumping out more beats, or in this case, more fat droplets. In Alzheimer's brains, DGAT2 sticks around way too long, basically hosting an all-night rave inside microglia. When the researchers used drugs to either block DGAT2's activity or kick it out of the cell sooner, the whole vibe changed. The fat levels dropped, microglia sobered up, and suddenly they were back on the job, eating plaques and restoring balance. That's what makes this discovery so refreshing. It's not about attacking plaques with an expensive drug and hoping for the best. It's about flipping a metabolic switch and letting the brain's natural defenses take care of business. It's the difference between being the person who has to clean the party yourself and being the person who just calls in the cleaning crew and watches them do their thing. Even better, this research suggests that lipid buildup could be the missing link in more than just Alzheimer's. Lipid droplet stuffed cells have been spotted in other neurodegenerative conditions in aging brains and even after traumatic injuries. That means this lipid party model could help explain why so many different brain conditions share a common theme of inflammation and immune cell dysfunction. If we can learn to tone down the party early, we might not just slow Alzheimer's, we could protect memory and mental clarity across a lifetime. And there's something weirdly optimistic about this whole idea. It means microglia aren't just broken beyond repair. They're still in there, capable of working, just distracted by their own lipid stash. The fact that scientists can give them a little metabolic reset and watch them spring back into action is a hopeful sign that we're not as powerless against dementia as we used to think. So yes, the lipids are partying, but that's actually good news because parties can be shut down. The bouncers can get back to work. And if researchers keep finding ways to restore microglial balance, we may end up with treatments that don't just slow Alzheimer's, but help the brain defend itself, year after year, plaque after plaque.